Hello everyone and welcome to our channel Mootechie. A warm welcome to all the dairy technologists and students with us today. We're beginning a new series with an introduction to the fascinating field of food microbiology. First, let's define our key terms. What is food? Food is a substance consisting essentially of protein, carbohydrate, fat, and other nutrients used by an organism to sustain growth, vital processes, and to furnish energy. Its absorption and utilization by the body is fundamental to nutrition and is facilitated by digestion. Next, what is microbiology? Microbiology is the study of microorganisms or microbes. This is a diverse group of generally minute simple life forms that includes bacteria, archaea, algae, fungi, protozoa, and viruses. The field is concerned with their structure, function, and classification, and with ways of both exploiting and controlling their activities. So what is food microbiology? Food microbiology is the scientific study of microorganisms, both those found in food and those used for the production of food. This includes microorganisms that contaminate food, as well as those used beneficially to produce foods like yogurt, cheese, beer, and wine. Moving on, we will see the factors affecting the kinds and numbers of microorganisms in food. The type of spoilage caused by microorganisms and enzymes depends on the kinds and numbers of these agents present and the environment around them. Most raw foods contain a variety of bacteria, yeasts, and molds, and may also contain plant and animal enzymes. The kind and number of microorganisms present on or in food are influenced by the kind and extent of contamination. Furthermore, environmental conditions can lead to a succession of organisms. One organism may initiate spoilage, and then one or more other kinds can produce secondary spoilage. Contamination can increase the number of microorganisms in food. For example, wash water may add surface bacteria to butter, and plant equipment may add spoilage organisms during processing. This increased bioburden, especially of spoilage-causing microbes, makes preservation more difficult. Conversely, pretreatment of foods may remove or destroy some microorganisms and inactivate food enzymes. Washing can remove surface organisms or add some from the water itself. Using an antiseptic or germicidal solution can greatly reduce microbial numbers, as can treatments with high temperatures raise ozone, sulfur dioxide, or germicidal vapors. Next, we explore factors affecting the growth of microorganisms in food, starting with associative growth. Associations between different microorganisms are involved in the spoilage or fermentation of most foods. Competition between bacteria, yeasts, and molds in a food usually determines which one will outgrow the others and cause its characteristic type of spoilage. If conditions are favorable for all, bacteria usually grow faster than yeasts and yeasts faster than molds. However, microorganisms are not always antagonistic. They can sometimes be symbiotic, meaning mutually helpful. Sometimes they are synergistic, where growing together they can bring about changes like fermentations that neither could achieve alone. For example, Pseudomonas cincinnati growing alone in milk produces a light brownish tinge, and Streptococcus lactis causes no color change. But when both grow together, a bright blue color develops due to the pH effect on the brown pigment from P. cincinnati. Another concept is metabiosis, where one organism makes conditions favorable for the growth of a second. Both may grow at the same time, but more commonly one succeeds the other. A classic example is raw milk at room temperature. It first supports an acid fermentation by Streptococcus lactis and coliform bacteria until they are inhibited by their own acid. Next, acid-tolerant lactobacilli increase the acidity further until they too are stopped. Then, film yeasts and molds grow on the surface, finally reducing the acidity so that proteolytic bacteria can become active. The effect of environmental conditions is paramount. The environment determines which microorganism will outgrow the others. The chief factors are the intrinsic or chemical properties and the extrinsic or physical properties of the food itself. Now, let's look at a table summarizing different groups of microorganisms and their association with foods. For bacteria, associations include foodborne disease, food spoilage, food fermentation, and the production of food additives and enzymes. For molds, associations are foodborne disease, food spoilage, production of single-cell protein, food fermentation, and production of enzymes. For yeasts, associations are food spoilage, food fermentation, and production of food additives and enzymes. For viruses, associations are foodborne disease, identification of food poisoning bacteria, and failure of dairy fermentations. Coming up, we discuss food spoilage. Food is considered spoiled when an undesirable change in its color, flavor, odor, or texture has occurred. The onset of spoilage is a gradual process that can happen due to poor sanitation, enzymatic or chemical reactions, improper temperature controls, microbial growth, or physical abuse. Foods can be classified by their ease of spoilage. First, Stable or non-perishable foods. These do not spoil unless handled carelessly and include products like sugar, flour, and dry beans. Second, semi-perishable foods. If properly handled and stored, these will remain unspoiled for a fairly long period. Examples include potatoes and apples. Third, perishable foods. This category includes most important daily foods that spoil readily, such as meats, fish, poultry, milk, eggs, fruits, and vegetables. Now let's examine the biochemical changes causing food spoilage. Most changes in foods are due to alterations in their biochemical properties by microbes, including carbohydrate, protein, lipid, or pectin degradation. Carbohydrate decomposition by microorganisms produces compounds like carbon dioxide, hydrogen, lactate, acetate, butyrate, ethanol, propanol, butanol, diacetyl, acetoin, butanediol, and dextran. Depending on the carbohydrate and the microorganism, the food may become slimy and develop an unpleasant taste or smell, becoming what we call stale. A table summarizes different types of fermentations. Alcoholic fermentation produces ethanol and CO2. Homofermentative produces lactic acid. Heterofermentative produces lactic acid, acetic acid, ethanol, and CO2. Propionic acid fermentation yields propionic acid, acetic acid, and CO2. Butyric acid fermentation gives butyric acid, CO2, and H2. Mixed acid fermentation results in lactic acid, acetic acid, H2, ethanol, CO2, butanediol, and formate. The most common of these are butyric acid, mixed acid, and butanediol fermentations. Protein decomposition involves microorganisms degrading proteins into compounds like CO2, H2, H2S, amines, keto acids, mercaptans, and organic acids. This process is called putrefaction, causing food to rot and produce a very bad smell. Protein degradation typically doesn't start until other nitrogen sources are lacking. Once it begins, microbes secrete proteases and peptidases to break down proteins into amino acids, and many peptides can contribute a bitter or sweet taste to the spoiled food. Lipid degradation, also known as rancidity, involves microbes breaking down lipids into fatty acids, glycerol, aldehydes, and ketones, producing bad smells and tastes. Lipid peroxidation requires factors like oxygen, unsaturated fats, light, high temperature, and metal ions. The resulting alcohols, ketones, and aldehydes impart unpleasant odors and tastes. Some fungi have lipoxygenases that oxidize unsaturated fatty acids to hydroperoxides and ultimately to aldehydes and ketones. This degradation can be avoided by reducing exposure to oxygen, adding antioxidants, and lowering the temperature. Finally, let's discuss pectin hydrolysis. Pectins are carbohydrates made of partially methylated polygalacturonic acid. They provide mechanical rigidity in fruits and vegetables. Pectin can be degraded by pectinases, which may activate during fruit ripening, making the fruit soft. Once mechanical damage occurs, microbial attack is often initiated. In general, the preference for spoilage is carbohydrates first, followed by proteins and lipids, though the amount and availability of the carbohydrate and the specific degrading microorganism are also crucial. We conclude with food poisoning. It's important to know that foods contaminated with pathogenic microorganisms usually do not look, taste, or smell bad. A foodborne infection is caused by ingesting food containing live bacteria that grow and establish themselves in the human intestinal tract. A food intoxication is caused by ingesting food containing toxins formed by bacteria that grew in the food. The live microorganism itself does not have to be consumed. Thank you for joining this introductory session on food microbiology. We've covered what food microbiology is, the factors influencing microbial growth in food, the different types of microbes involved, the processes of spoilage, and the basics of food poisoning. This foundation is essential for anyone working in dairy technology and food science. Thank you. See you in the next class.